So in today's video, we're going to be discussing apoptosis, which is the cell's controlled death cycle. By the time this video ends, you're going to understand completely what apoptosis is, physiologic roles of apoptosis, morphologic changes that are seen in apoptosis, what triggers it to happen, clinical relevance, and at the end, we're also going to throw in some USMLE practice questions, not only to test your knowledge, but more importantly, solidify your knowledge for the long term. Don't forget to check out thehealingpowerofnature.com for the best resources on the market and also our Patreon. Apoptosis. This is the cell's built-in self-destruct system. Some etymology you need to be aware of. So the words apo means away and tosis means falling. So it's illustrating the organized process where cells fall away, similar to how leaves fall away from a tree. Contrasting necrosis, which is chaotic and leads to inflammation, while apoptosis is ATP dependent, precise, and clean. So some definitions: apoptosis is programmed cell death that requires ATP and also gene activation. Targets individual cells or small groups of cells, avoiding large-scale tissue destruction as seen in necrosis. As I mentioned, there is no inflammation that occurs. It's due to rapid clearance of apoptotic bodies by phagocytic cells like macrophages. Apoptosis is indispensable for proper development, immune regulation, and cancer prevention. Dysregulation of apoptosis underlines many diseases, including cancer, autoimmune disorders, and also neurodegeneration. Some examples of physiologic apoptosis: embryogenesis and organ development. So it actually forms the digits. Apoptosis shapes the fingers and toes by removing integrated tissues. There's also neuronal pruning. Apoptosis removes excess neurons, ensuring optimized brain wiring. There's also hormonal dependent tissue remodeling. For example, during menstruation, endometrial cells undergo apoptosis. Post-lactation breast tissue apoptosis reduces the number of milk-producing cells when breastfeeding stops. Additionally, there's immune system maintenance. Negative selection of T cells in the thymus. Self-reactive T cells undergo apoptosis to prevent autoimmune disease. CD8 plus T cells induce apoptosis in virus-infected or cancerous cells via perforin and granzyme release. Also, DNA damage response and cancer prevention. So p53-mediated apoptosis, the tumor suppressor gene p53, or the molecular policeman, detects DNA damage. If damage is beyond repair, p53 induces apoptosis. This prevents mutations from leading to cancer. It also prevents uncontrolled proliferation of cells with damaged DNA, which is a hallmark of tumorigenesis. A key mnemonic you can remember for apoptosis is AID. A, autoimmune prevention, T cell selection in the thymus. I, for infected cell removal by CD8 T cell induced apoptosis. And D, DNA damage repair and cancer prevention by p53 tumor suppression. So what it looks like under a microscope: apoptosis can be identified under a microscope through distinct morphologic changes that differentiate it from necrosis. So key morphologic changes you'll see is 1, cell shrinkage. The cytoplasm becomes dense and contracts. Unlike necrosis, where the cell swells, this shrinkage makes the cell appear smaller and more compact. There's also increased eosinophilia. The cell stains pinker on H&E stains due to protein condensation in the cytoplasm. There's also nuclear changes, specifically, like we've talked about in other videos, 3 distinct steps. There's pycnosis, where the nucleus shrinks and becomes darkly stained because of condensed chromatin. There's karyorrhexis, where the nucleus fragments into smaller pieces. Karyolysis is absent because there's no nuclear fading, unlike necrosis. Formation of apoptotic bodies. The cell fragments into small membrane-bound bodies contain nuclear and cytoplasmic material. These bodies are intact and prevent leakage of cellular contents. There's also no inflammation. Apoptotic bodies are quickly engulfed and digested by macrophages, preventing an immune response. A good mnemonic you can use to remember this is safe death. What does this mean? There's shrinkage. Apoptotic bodies form fragmented nucleus via pycnosis to karyorrhexis, and then E, they're engulfed by macrophages. 3 major pathways you need to be aware of. First is going to be the intrinsic or the mitochondrial pathway. This is the cell's internal suicide switch. So what happens is there's a cellular stress like radiation, toxins, or hypoxia, DNA damage, which activates p53, preventing replication of damaged cells by initiating apoptosis, or growth factor withdrawal, apoptosis of endometrial cells during menstruation. The key molecular event is BCL2 inhibition. So pro-apoptotic proteins like BAC inhibit anti-apoptotic BCL2. This allows mitochondrial permeability. Once there's mitochondrial permeability, cytochrome C leaks from the mitochondria into the cytoplasm. Formation happens where cytochrome C, this activates capsases, the executioner enzymes. A key mnemonic is BCL2 blocks cytochrome C leakage. BCL2 prevents cytochrome C leakage. When BCL2 is inhibited, cytochrome C leaks. This triggers apoptosis. An example is seen in radiation therapy. This induces apoptosis in cancer cells by causing a DNA damage, or additionally endometrial apoptosis after progesterone withdrawal during the menstrual cycle. There's the extrinsic or death receptor pathway. This is the external assassination signal. There's two major mechanisms. First is the fast ligand pathway, also known as CD95. What happens is the fast ligand binds to the fast receptor, CD95, activating capsases, activating apoptosis. An example is a negative selection of self-reactive T cells in the thymus to prevent autoimmunity. The other pathway is TNF alpha, tumor necrosis factor alpha pathway. What happens is tumor necrosis factor alpha binds to TNF receptors. This activates capsases, which activates apoptosis. An example is cytokine-induced apoptosis and inflammation. So remember, fast equals fast death. Fast binds the fast ligand, triggers rapid apoptosis, especially in immune regulation. Lastly, there's cytotoxic CD8 T cell pathway. This is the assassin mechanism. What happens is CD8 positive T cells recognize infected or cancerous cells displaying abnormal antigens on MHC1 molecules. It releases two proteins, perforins, which poke holes in the target membrane, and granzymes that enter through perforin pores and activate capsases. Capsases then activate apoptosis. Remember, CD8, cytotoxic destroyer, perforin and granzymes. An example would be in viral infections. CD8 T cells kill virally infected cells. Some key USMLE takeaways are to make this as simple as possible. The three different pathways. First, you have the intrinsic pathway, internal damage, BCL2 inhibition, cytochrome C releases, apoptosis happens. Extrinsic pathway, the death receptor signal, fast ligand or TNF alpha binds receptor, leading to what? Apoptosis. Or the CD8 T cell pathway, which direct cytotoxic killing. Basically, perforin and granzymes are activated, which then activates apoptosis. Here's a simple chart that I made for you guys. I would recommend just going ahead and taking a picture of this, keeping it close. We're going to our website to get it downloaded. Basically, you have apoptosis or necrosis. Apoptosis requires energy. The cell shrinks. There's no inflammation. It's clean. It's capsase mediated. Causes DNA damage, hormonal withdrawal, or infection can cause apoptosis. Examples are CD8 T cell killing or endometrial shedding. Whereas with necrosis, there's no energy is required. It's passive, it's accidental, it's caused by an external factor. The cell swells, there's inflammation, it's uncontrolled cell rupture, so enzymes leak into the bloodstream. It's caused by trauma, ischemia, toxins, or infections. Examples include myocardial infarction, stroke, pancreatitis, or gangrene. Clinical relevance. Too much apoptosis equals excessive cell death. So in neurodegenerative diseases like Parkinson's and Alzheimer's, loss of neurons due to inappropriate apoptosis. Also in aplastic anemia, bone marrow progenitor cells undergo apoptosis, leading to pancytopenia. Or too little apoptosis equals cancer growth. So BCL2 overexpression in follicular lymphoma. BCL2 prevents apoptosis, uncontrolled B cell survival, lymphoma. Or also in HPV E6 and E7 proteins. HPV suppressors, P53, the molecular policeman, blocks apoptosis, viral persistence, and cancer risks. Let's go ahead and jump into a few practice questions. I'll always advise you to read the last sentence first, then read your question responses, then read the entire question. I'll go ahead and give you a few minutes and then we'll go ahead and discuss the problem. So the correct answer was cytochrome C, or letter C. The explanation for this is chemotherapy induces DNA damage, triggering the intrinsic or mitochondrial pathway of apoptosis. Let's go ahead to the next question. I'll give you a few minutes and then we'll come back and discuss the problem. So if you chose B, that is correct. The fast ligand pathway is part of the extrinsic apoptotic pathway and plays a critical role in the negative selection of T cells in the thymus. During negative selection, T cells that recognize self-antigens undergo apoptosis. So let's do one more question. I'll give you a few minutes, then we'll come back and discuss the problem. So if you chose D, that is correct. The release of peripheral granzymes. So basically, CD8 positive T cells induce apoptosis in virus-infected cells like hepatocytes and hepatitis B via the cytotoxic pathway. So that concludes our video for today. If you found this video to be helpful, be sure to check out thehealingpowerofnature.com and also our Patreon for some of the best study materials on the market. Thank you very much and see you next time.